Hello students, welcome to all these further classes. So today I will be taking the third part of the chapter, the fundamental unit of life. It is the fifth chapter from class 9 science CBSE board. Okay, so we have discussed a lot in my past two videos. If you will go, you will find it that we have discussed about many cell organelles. Okay, and we have discussed about many things about cell. But here some of the cell organelles were left, which we are going to discuss today in the part 3. So today I will be discussing about vacuoles. Okay, the type of vacuoles. In the type of vacuoles, we shall be discussing about all the three types of the vacuole. That is the sap vacuole. Okay, the contractile vacuum. And also the food vacuum. Okay. Then we shall see the osmoregulation in amoeba, that how amoeba maintain its water content. Okay, osmoregulation is done in amoeba, a pictorial representation of how it maintains its water we will be seeing. Then we shall see the difference between the plant cell and animal cell. The vacuole of the plant cell and animal cell are different. So we shall be seeing the difference between that, and it is very commonly asked in exams. Next we shall see about centrioles, how these centrioles they help in cell division. Okay. Next comes the ribosomes. We shall be seeing about ribosomes. Then we will see about peroxisomes, how it helps in enzyme digestion. Digestive enzymes are present in it. Okay. In the last, we shall be discussing about the difference between the prokaryotes and eukaryotes. This is also very important from examination point of view. And next, the difference between the animal cell and the plant cell. Okay, so students, the differences between the plant and animal vacuum, the difference between the prokaryotes and eukaryotes, and the difference between the animal cell and plant cell. Out of these three, any one is surely asked in your exams. So you must see it carefully, you must see the difference between them carefully. So we shall be starting with the vacuoles. So let's start. So the vacuoles, they are the storage sacs. Okay, what are they? They are storage sacs for solids or liquids. Okay. So what does this storage sacs means? Solid for solid or liquids content. It means that there is a place where these cells they can store their solid and liquids. Okay, so vacuole is a place where cells can store their solids and liquid contents. Okay, so these vacuoles they are present in both. Animal cell and plant cell. Okay, so they are present in animal cell and plant cell both. The only difference is that the plant vacuoles are bigger as compared to the animal vacuoles. Okay, so plant vacuoles are bigger in size than animal vacuoles. Okay, so they are much bigger. They can be means in plant cell it is around, in some of the cases you can see that these vacuoles they form around, or they can occupy around 50 to 90% of the cell volume. Okay, how much volume? Okay. So there are basically three types of vacuoles. Okay. The three types of the vacuoles are, the first one is the sap vacuole. Okay. The second one is the contractile vacuoles. Contractile vacuoles. Okay. And the third one is the food vacuoles. Okay. So let's see about sap vacuoles. So what happens in plant cell? The vacuoles are full of sap vacuoles. Okay. They are full of the sap vacuoles in plant cells. And what did they do? The function of the sap vacuoles is that they provide turgidity and rigidity to a cell. And rigidity to plant cell. Okay. I am talking about plant cell. So in plant cell, the vacuoles are full of sap vacuoles, and the function of the sap vacuole is to provide turgidity and rigidity to the cell. Okay. In plant cell. Now what happens? Many substances of importance, okay, in the life of a plant cell are present in the sap vacuole. Okay, in this sap vacuole, many substances that are important for the plant cell are present in it. For example, there are those substances which are present are like amino acids. Okay. Sugars are there. Okay, these all are important for plant cells, and these are present in this sap vacuoles. Okay, various organic compounds, compounds, or you can say various organic acids. In fact, you can better it's to say various organic acids. Proteins. Okay, so these important substances are present in the sap vacuoles. Many important substances which are important in the life of a plant cell are present in the sap vacuoles. Now what does sap vacuole do? It gets separated from the cytoplasm. Okay, how the sap vacuole is separated from the cytoplasm? By a semi-permeable membrane, and that membrane is called as tonoplast. Okay, please listen this carefully. The term tonoplast is very common and is mostly asked. So what is tonoplast? The sap vacuole are separated from the cytoplasm. Tonoplast is a separation between sap vacuole, okay, sap vacuole and the cytoplasm. Okay, so if you are asked in your exam what is a tonoplast, so you can say that it's a semi-permeable membrane which separates the sap vacuole and the cytoplasm. Okay, so the main function of the sap vacuole is that they allow a rapid exchange between cytoplasm and the surrounding environment. ठीक है, जो surrounding environment है और जो cytoplasm है, इनके between cytoplasm and the surrounding. Okay. So the number of sap vacuoles are found in the young cells and animal cells. Okay. So number of the sap vacuoles are found in the young plant cells and animal cells. What happens in mature plants? A small sap vacuole combine together to form a large central vacuole. Okay. So in young plant this sap vacuole is present, but what happens in a mature plants, the plants that are grown up or that are mature, the small sap vacuoles they combine together. All the sap vacuoles, small, they combine together and form a one big central large vacuole. ठीक है? सारे छोटे-छोटे sap vacuoles combine करके एक बड़ा central vacuole बना देते हैं. Okay? So this was about sap vacuoles. I hope it must be clear to you. Now the second type of vacuole is a contractile vacuum. So what is this contractile vacuum? These contractile vacuoles they are found mostly in protistants. Okay. Mostly they are present in protistants and also in algal cells. Okay. Algae में पाया जाता है. Algal cells. Fine. So these protistants or these algal cells they live in a fresh water. So what happens? The membrane of this contractile vacuole is highly extensible and collapse easily. क्या होता है कि जो membrane है ये जो sap vacuole की ये extend हो सकती है, बहुत extend हो सकती है. Okay? और इसकी वजह से क्या हो जाता है ये collapse कर जाती है. It collapse easily. Okay? कोई भी चीज जो extend हो सकती है, like आप balloon को लीजिए, balloon को आप blow करिए, balloon gets extended and extended, and a stage comes when you fill much of the air in a balloon, that balloon burst. Okay? Balloon collapse कर जाता है. Why is that? Similarly is the case in the contractile vacuoles. They have a capacity to get extended when you okay, and a stage will come that after a lot of extension they collapse. Okay. So these contractile vacuoles, what is their importance? They play an important role in expelling excess water. So what is this? Expelling excess water. Okay. Their role is in expelling excess water. So this expelling excess water means osmoregulation. Okay. They play an important role in osmoregulation. What happens in osmoregulation? It means that these contractile vacuoles they help the cell to maintain their water content. Okay? किसमें ये help करता है? Excess water को expel करता है, osmoregulation में help करता है. Means कि वो जो भी water का जो water content है उसको मतलब maintain करने में वो help करता है. It helps in maintaining the content of the cell. And also one important function of this contractile vacuole is that the waste materials from the cell. They expel the waste material that are present in the cell. Okay? So now I will be showing you a diagram how this amoeba they perform the osmoregulation. Okay? So let's see osmoregulation in amoeba. Okay? So here you can see this is a figure in which these are the steps how osmoregulation takes place in amoeba through contractile vacuum. As I have already told you, osmoregulation means exhaling of water, means the cell need to maintain the water content. Okay? Maintenance of water content in the cell. So in the first step, this is the amoeba. Okay? So in the first step, you can see that this is a contractile vacuum. Okay? This one is a contractile vacuum. So water enters in the contractile vacuum through a process of osmosis. will burst, it will release all the water from it. Okay, and after that this amoeba is going to start the process again. Okay, amoeba gets reshaped and again this process will continue. In fact, you can draw an arrow here also, that now again water will enter through osmosis in the contractile vacuum. This contractile vacuum is going to swell and further it is going to burst to release the water, and amoeba will attain the previous position again and again. This cycle keeps on continuing. So this was about the osmoregulation in amoeba through contractile vacuum. Okay, so this was about contractile vacuum. We have seen about stap vacuoles, contractile vacuoles. Now we will see about food vacuoles. Okay, so the third type of the vacuole is a food vacuum. Let's see. So the third type of a vacuole is a food vacuole. Okay, so this type of vacuole is also found in amoeba and all. They are found in the cells of protozoans and some lower animals. Okay, in protozoans and lower animals. Fine. So it is found in protozoans and in lower animals. What happens? This food vacuole, it is responsible for the digestion of food. What is the function of food vacuole? Digestion of food. Okay, digestion of food in the cells. Okay, digestion of food in the cells. You can write here in the cells. How does digestion takes place in the cells? Because there is food vacuoles, they contain digestive enzyme with them. Okay, so they contain food digestive enzymes. So that why they help in the digestion of food. What they contain? They contain food digestive enzymes. Fine. Now what happens? This digested food is then passed into the cytoplasm. Okay, this digested food. Digested food is passed into the cytoplasm. Okay, it is passed into the cytoplasm or found in most of the single cell organelles like amoeba. Okay, so this was about the food vacuoles. We have seen stap vacuoles, contractile vacuoles and food vacuoles. Now we shall be seeing the difference between the animal vacuole and a plant vacuole. Okay, so let's see. So let's see the difference between the plant cell vacuole and animal cell vacuoles. Okay, so we can see that plant cell vacuole stores all material that is required for the plant to stay alive. Okay, so what they store? They store materials like water. Okay, but if we talk about animal cell vacuoles, these animal cells, they contain food items in unicellular organism. Okay, so they store all materials that is required and they contain only food items. Then the second thing is that plant vacuoles maintain the turgidity of the plant cell. Okay, this is because of the plant vacuoles present in a plant cell, the turgidity of the plant is maintained. But in case of animal vacuoles, can expel expel water and waste out of the cell. Okay, we have already seen the examples I have already explained the examples. So animal vacuoles can expel water and waste out the cell. Now the next is that plant cells generally contain a single large vacuole. Okay, so in plant cell a single large vacuole is present, and these vacuoles in some of the plants are so big that they cover at least 50 to 90% of the cell cytoplasm. Okay. Then if we talk about cell animals, they contain several small vacuoles. Okay, in animal cell there is a small many vacuoles present, but in plant cell only a single one vacuole is present. Okay. Now the last one is that plant vacuoles are present in the center of the cell. Okay, but in case of animal vacuoles they are scattered throughout the cell. It is not that they are present in the center, but they are scattered throughout the cell. Whereas in plant vacuoles, they are confined to the center. Okay, so this was the difference between the plant cell and animal cell vacuole. Next, we shall see about centrioles and all. So next, we shall see the centrioles. So what are centrioles? Centriole is a small set of microtubular arranged in a specific way. Okay, so they are arranged like this. Okay, microtubular, and it's a whole set of this microtubular arrangement. Like this, they are just arranged like this. More thin they are made little broader. So their main purpose is to help the cell in cell division. Okay. So the main function of centriole is in the cell division. Okay. Cell division. They are found near the nucleus, but they can be seen only during the cell division. Okay, although they are present near the nucleus, but they can be seen only during when the cell divides. Okay, so and what the centrioles, they are present in a pair. Okay, they are present in a pair like this. Okay, they are present in a pair and they form a spherical substance that is called the centrosome. What they form? A spherical substance that is called as centrosome. Okay, which appears near the nucleus. That also appears near the nucleus, and this centriole is also present near the nucleus. So what happens when this cell division takes place? These centrioles, they also divide. Okay, they divide into two parts, and each part moves to the opposite side of the cell. Okay, they divide into two parts and each cell moves to the opposite part of the cell. Okay, so this was about centriole. Now let's see about ribosomes. I have already discussed about ribosomes in endoplasmic reticulum. That endoplasmic reticulum is of two types, soft and rough. So if the ribosomes are attached to it, it's a rough one, and if it is not attached, it's a soft one, a smooth one, sorry. So here we shall be seeing some of the key features of a ribosome. Okay, so they are also cell organelles, and their main function is the protein synthesis. They are responsible for protein synthesis. Okay. Then the next thing is that these ribosomes, they can be found in both. They are present in the prokaryotes as well as in eukaryotes. Okay. They are present in prokaryotes as well as eukaryotes. Okay, so their main function is protein synthesis. They are present in prokaryotes as well as in eukaryotes. So what happens in prokaryotes? These ribosomes, they float freely in the cytoplasm. Okay? In case of prokaryotes, they float freely. Where they float freely in the cytoplasm. Okay? And what happens in case of eukaryotes? This was for prokaryotes. In eukaryotes, they can be found floating in the cytoplasm, or they can often be attached to the endoplasmic reticulum. As we have already discussed that if these ribosomes, they are attached to an endoplasmic reticulum, they form a rough endoplasmic reticulum. RER, rough endoplasmic reticulum, means these ribosomes are attached. Ribosomes are attached to the endoplasmic reticulum, or they can float free in the cytoplasm. Okay. So next thing about the ribosomes is that the ribosomes that are attached to the endoplasmic reticulum, they help mainly in the protein synthesis. Okay. What they do, they are able to export. What they do generally, they protein synthesis that are to be exported out of the cell, while the ribosomes, they are floating inside the cell. Okay, so this protein synthesis are used inside the cell. Okay, so this was about ribosomes. Next cell organelle which I will be taking is peroxisomes, and this is the last cell organelle which we are going to discuss. Okay, so next one is the peroxisomes. Okay, this is also a cell organelle. So peroxisome is a small vesicles found in the cell. They are very small, minute type. Okay, so the enzymes, these enzymes are used to break the toxic material inside the cell. So what they do, enzymes break the toxic material inside the cell. Okay? Then what happens ki next is what? They also helps in the digestion of fatty acids of the cell. What they do? They digest fatty acids of the cell. Okay? And digest fatty acid of the cell, as well as they also that amino acids also, by carrying out what they do, they carry out oxidation reaction in the cell. Okay, the digestion of fatty acid of the cells and amino acids. Okay, so what they do? By the oxidation of the cell. Okay, they carry out oxidation reaction in the cell. And they are also responsible for the digestion of alcohol in human body. Okay, digestion of alcohol in human body. So what happens is they help in the digestion of alcohol in human body. So our liver in human body, the liver contains much of these large number of peroxisomes. Okay, that's why it is said that if you drink lots of alcohol, it affects your liver only, because this peroxisome, the enzyme which is digesting the alcohol, is present in liver only. Okay, so this was about all the organelles which were left. We have not discussed in our previous videos. So here we finish the organelles. Next we shall be seeing the difference between the animal cell and plant cell. So till now we have studied all the cell organelles. So before starting the difference between the plant cell and animal cell, let's see what are the difference between prokaryotes and eukaryotes. Okay, so in prokaryotes, nucleus is absent. Okay, nucleus is absent here, whereas in eukaryotes, nucleus is present. Fine. In prokaryotic cells, the nuclear membrane is absent. Since there is no nucleus, so nuclear membrane is also going to be absent. Okay, so prokaryotic cell, they lack nuclear membrane. Okay, whereas eukaryotic cell have nuclear membrane. Okay, so since they don't have nucleus, there is a no nuclear membrane. Since they have nucleus, there is a nuclear membrane also. Okay, so these prokaryotic animals, they have a single chromosome. Which type of chromosome they have? They have only single chromosome. Whereas in case of eukaryotes, some animals have a single, or some may have multi-chromosomes. Okay? Multi-chromosomes. Fine. The prokaryotes are very primitive. Okay, primitive cells. So what happens? These prokaryotes, they only undergo with asexual reproduction. Okay. Whereas the eukaryotes, they are the developed cells, so they can undergo asexual as well as asexual both reproduction. Okay, so asexual or sexual reproduction. Mostly they have a sexual reproduction. Okay. So these are very primitive forms, okay? And if we see about the organelles, type of organelles, they are only unicellular. Means they are made up of only one cell. Okay? These are unicellular organelles, whereas the eukaryotes are multicellular organelles. Means they are made up of many cells. Okay? They are made up of only single cell, they are made up of many cells. Okay? Uni means one and multi means many. Okay? And now they don't have nucleus and nuclear membrane. Similarly, they don't have a membrane around the cell organelle. Okay? No membrane around cell organelles. Okay, cell organelles are also not having any membrane, and they have membrane around the cell. Okay, membrane is present around the cell. So you can say cellular membrane Okay. Next is that in case of prokaryotes, centriole is absent. Just now I have told you about centrioles. So centrioles we have just studied that they help in cell division. So in this case, in prokaryotes, centriole is absent. Okay. But if we talk about these eukaryotes, in eukaryotes the plant cells, the animal cells, not the plant cells. Centriole is present in animal cells. Centrioles is present in animal cell, and this helps the cell to divide. Cell division is because of these centrioles. Okay. So if we talk about the type of cell division which takes place in prokaryotes, so cell division is binary fission. Which type of cell division takes place? Binary fission. Okay. And in case of eukaryotic cell division is mitosis. Okay, it can be mitosis or it can be meiosis. Okay, both type of cell division takes place, mitosis and meiosis. Whereas in this only binary fission type of cell division takes place. Okay, so this was the difference between prokaryotes and eukaryotes. The main difference is that nucleus is absent, nucleus is present. If there is no nucleus, no nuclear membrane, nucleus is there, so they have a nuclear membrane. They have a single chromosome, they are multi-chromosomal. Asexual reproduction, they have sexual as well as asexual reproduction. Okay. Then they are unicellular, made up of single cells. They are made up multicellular, made up of many cells. No membrane cell organelle are not covered in this cell. Cellular membrane is present. Like you can take the example, membrane bound cells are there. Like mitochondria, there is a membrane. Centrioles is absent since there is no cell division, so there is no use of centrioles. But here cell division takes place, mitosis and meiosis. So cell divides, so centriole is present. Okay. So now let's see the examples. If we see the examples of prokaryotes, it's bacteria or cyanobacteria is there. Cyanobacteria means blue-green algae. Okay. Here the example is fungi is there, plant cell and animal cell. Okay. So this was the difference between the eukaryotic cells and the prokaryotic cell. Okay. So we have seen the difference between the eukaryotes and prokaryotes. Next we shall see the difference between the animal cell and plant cell. So in animal cell, cell wall is absent. Cell wall is the outermost covering of a cell, or you can say the outermost layer of a cell. So in case of animal cell, the outermost layer is the cell membrane. Okay, there is no cell wall. Okay, but cell wall is present in plant cell. So in case of plant cell, the outer covering of the cell is the plant cell, and then comes the cell membrane. But in case of animal cell, cell membrane is the outer layer of the cell. Okay. Now plastids are absent. Okay. If we talk about plastids, it is absent in plant cell, animal cell. Okay. But in plant cell, plastids are present. Plant cell, these plastids that are present in the chlorophylls, they help in photosynthesis. Means this in making the food. Okay. Next is that in case of animal cell, centrioles are present. Okay. Centrioles are present because in animal cell the cells divide. Okay, there is a division of cell. So centrioles are present. Centrioles help in the cell division, whereas centrioles are absent in case of plant cell. They are present in the animal cell. Okay. Next comes that Golgi bodies. Okay, we have already studied about Golgi bodies. It is an organelle of a cell. It is present in plant cell. Animal cell. Golgi bodies are present. Okay. In animal cell, it is present. But in plant cell, what happens? It is present. It is not that Golgi bodies are not present. They are present. But they are called as dictyosomes. Okay. Golgi body is present and it is called as dictyosome. Okay, so this is a Golgi body or Golgi apparatus, also it is called. It is a cell organelle which is also called as the suicidal bags. Okay. So in this case, animal cell vacuoles are either absent, or if vacuoles are present, they are very small. Small vacuoles are found. Okay. So vacuoles are small. Either they are absent, or if they are present, they are very small. But in plant cell, vacuoles are large. In fact, you can say that they cover 50 to 90% of the cytoplasm. Okay? And in animal cell, vacuoles are many vacuoles can be there, small, but in plant cell they are big one vacuole. Okay? So in this, centrosome is present, whereas in plant cell it is absent. Okay? Centrosome is present. We have just studied about centrosomes. In this video only I have studied, and 2 centrioles are there. But in this case, the centrosomes are absent. Okay? So this was about the difference between the plant cell and the animal cell. That cell wall is absent, cell wall is present, plastids are absent, plastids are present, which helps in photosynthesis. Centrioles are present, that helps in cell division, but it is absent in this case. Golgi bodies are present. In this, Golgi bodies are present, but are called as dictyosomes. In this, vacuoles are small vacuoles are there. In this, one large big vacuole is present. Centrosome is present and centrosome is absent. So this was about the difference between the plant cell and animal cell. So here I am going to finish the third part of the chapter, the fundamental unit of life. Here we have seen some organelles along with eukaryotic and prokaryotic difference, and the difference between the animal cell and the plant cell. So thank you for watching my video. If you like it, please give a like button, and if you have not subscribed our channel yet, please do subscribe Always Scholars so that you can get the daily updates of the video. We put the video daily, and we have also developed our app. You can just go on Google app. From there you can download the app. Okay, and in the app you will find very interesting videos that is going to help you in learning the chapters. Concept wise videos are there, topic wise it is there, notes are there, PDF notes which are according to the CBSE board topics, and if you will see there are some MCQ questions which you can just solve it and see that how much you are able to do. And if you are interested for the live class, we have one-to-one teacher and student interactive classes. For that, you can just mail us on our Oli Scholar mail ID. So thank you so much. Thank you for watching my video. Thank you.